Let's start this video with a question. What would happen if continuous self-pollination occurs in flowering plants? The question says that if continuous self-pollination or inbreeding occurs in flowering plants, what would happen? What it would be leading to? You may be coming up with different question. I would prefer to give this as answer. Occur inbreeding depression. We already discussed about inbreeding depression. So you know that inbreeding depression is reduction in fertility. Reduction in fertility here refers to decrease in fertility or decrease in ability to reproduce. The plant is becoming sterile. If the plant is losing its ability to reproduce, then the productivity will be reduced. The plant won't be able to reproduce efficiently. When it comes with reference to pollination, we can see that there is reduction in pollen production. If there is reduction in pollen production, then gradually there is decrease in production of seeds, and the seeds which are produced are having low quality and the quantity of seeds produced will be low. So inbreeding depression will arise whenever continuous self-pollination occurs for say up to 6th generation, 4 to 6th generation. If there is continuously self-pollination or inbreeding is occurring, then inbreeding depression occur. The term depression here refers to decrease in something. What is being decreased? The fertility of the plant is being decreased. If the plant is becoming sterile, there is losing of ability to reproduce, and which in a way it leads to extinction of the plant, as the plant won't be able to survive. So without discussing much, what would be the solution for inbreeding depression? Usually the plants themselves naturally employ some mechanisms or technique in order to solve the problem of inbreeding depression, and that is outbreeding devices. The devices here refers to technique, mechanism, adaptations, or different ways or different methods which are being employed by flowering plants in order to avoid inbreeding depression, in order to avoid self-pollination so that they can undergo cross-pollination. So this video is to discuss about outbreeding devices as part of chapter number 2, sexual reproduction in flowering plants. First, let us make ourselves clear about the topic outbreeding devices. What exactly is outbreeding devices? Out refers to not inside, right? The plant is undergoing crossbreeding. Outbreeding here refers to crossbreeding or cross-pollination. So devices, outbreeding devices here refers to, these are mechanisms or techniques or ways, methods, or these are adaptations in flowering plants which usually prevent or discourage self-pollination, and which in a way it will be the plants will be encouraging cross-pollination, mainly for becoming more fertile so that they can increase their genetic diversity. So outbreeding devices are mechanisms which usually prevent self-pollination and which helps in encouraging cross-pollination. And these are usually barrier to self-pollination, as we had discussed. And outbreeding devices usually employ or usually make use of external pollinators. so that they can undergo cross-pollination. Why do flowering plants opt for outbreeding devices though it is uneconomical? Self-pollination is easy to occur, but why flowering plants are making use of outbreeding devices by spending energy, by spending lots of resources, mainly to avoid inbreeding depression, so that they can undergo cross-pollination, which in a way leading to more genetic diversity, and they are going to survive more by producing fertile offsprings. Now, we have different uh, outbreeding devices which are usually uh, employed by flowering plants. Say dichogamy, we have heterostyly, unisexuality or diclyding, dioecy of plant, self-incompatibility, Polenprepotency. So these different outbreeding devices usually prevent self-pollination and helps in cross-pollination, which usually encourages cross-pollination. We'll discuss one by one. The first outbreeding device which is used for preventing self-pollination or inbreeding is dichogamy. Di here refers to two. Dichogamy, gamy here refers to fusion. In dichogamy, usually pollen grain maturation and maturation of ovary occurs at different time. If pollen grain and ovary are maturing at different time, the release of pollen grain and stigma receptivity are not synchronized. Not synchronized here refers to not occurring at same time. If pollen release and stigma receptivity are not occurring at same time, then there is no possibility of occurring self-pollination, and they will be employing cross-pollination. Two different condition or mechanisms of dichogamy are prot-andry and protogyny. Pro here refers to early, andry here refers to male, and gyne here refers to female. So in prot-andry, there is maturation of pollen grains, or there is release of pollen grains earlier than stigma becomes receptive. As you can see in this cartoon, you are able to see release of pollen grains by anther of stamen. But the stigma is not receptive. Stigma is still immature. So this in a way prevents self-pollination, and the pollen grains which are released will be used by pollinators or will be cross-pollinated. On the other hand, in case of protogyny, there is immaturation, or the anther becomes immature and there is no release of pollen grain, but the stigma is becoming receptive. The stigma is receptive and it is not receiving its own pollen grains as the anther is not matured and there is no release of pollen grain. Instead of that, there is a possibility of receiving external pollen grains. So in a way, it leads to cross-pollination. The second type of outbreeding device is heterostyly. Hetero refers to different. Styly refers to the style of pistil. We have pistil here, so pistil is having stigma, style and ovary. So heterostyly here refers to the length of the style is different. Say, or on the other hand, we can also say that anther and stigma are placed at different position due to difference in length of the style. If the style is longer, then the anther will be shorter. If the style is shorter, then the anther will be growing at a distant place, will be growing far away from the stigma. This is usually observed in primroses. Primroses are having two types of flowers. We have trump flower and pin flower. In case of trump flower, you can see that the style is shorter. Shorter style is found in trump flower. In case of pin flower, we are able to see a longer style. The style is long so that anther is at the halfway point of the flower. Stigma is exposed near to the petals. But in case of trump flower, stigma is at the halfway point of the flower, and the anther is usually longer. Anther is usually placed far away from the stigma. Stigma is at halfway point of the flower, which in a way avoid self-pollination and favors cross-pollination. The third outbreeding device is dicliny or unisexuality of flower. Di refers to two, cliny here refers to bed or sofa bed. So usually plants are having two different flowers, unisexual flowers. Plants may be having staminate flower, that is male flower, and pistillate flower, female flower. And there is no bisexual flowers growing in those plants which are favoring diclining or unisexuality of flower as a mechanism of outbreeding device. For example, in case of castor or castor, usually Americans say castor, and we usually call it as castor, right? Castor, in case of castor, as you can see here, the pistillate flower is the female flower, and it is growing at different position in comparison to the staminate flower, which is the male flower. Both the female flower and male flower are growing far away from each other, which usually prevent self-pollination and favors cross-pollination. Same in the case of maize or corn, there are occurrence of two flower. Pistillate flower, which is female flower, have usually represented by cop. Cop is the structure which usually is the product of corn plant, and from cop, in the cop we used to have the corn seeds, and corn seeds are usually used for preparing corn flakes. We also consume eat seeds of corn as sweet corn, right? So male flower is the staminate flower, which is usually represented by tassel. So presence of male and female flower in case of corn plant and in case of castor prevent self-pollination and helps in cross-pollination. Now these are three outbreeding devices. The fourth one that we are going to discuss is Herkogamy. Herkogamy is a natural physical barrier which is a mechanism of separating pollen grains from ovary by not allowing the pollen grains to enter. So there is separation of pollen grains from ovary by preventing entry of pollen grains into the ovary. How it is prevented? Herkogamy usually occurs in case of orchids. In case of orchids plant, they are usually having a structure called pollinia, which is aggregate or assemblage or combination of many pollen grains which are attached together, which are bind together collectively. And that collective structure is called as pollinia. As you can see here, pollinia is plural and pollinium is singular. This is a real picture of orchid flower having pollinium, and the pollinium here is shown in golden color, and here you can see an enlarged image of pollinium. Pollinium is nothing, it is cohesive masses of pollen grains, or these are aggregate aggregation of pollen grains or combination of many assembly of many pollen grains and forming a structure that is called as pollinium, and pollinia is plural form. So such kind of structure prevent self-pollination as the pollen grains are stick together or hold together as pollinium or pollinia. So this is also this is a physical barrier or separation of pollen grains from entering into the ovary. Heterostyly is also a kind of Herkogamy. In heterostyly, there is separation of anther and stigma on the basis of length, difference in length of the style. So Herkogamy is physical separation of pollen grains from ovary. So self-pollination is prevented, and we have already discussed about heterostyly, which is also a kind of Herkogamy. Now the fifth outbreeding device is diec. Diec is existence of male plant and female plant. So the male plant will be growing male flower and female plant will be having female flower. The best example that we can give is papaya plant. Papaya plant are of two types, male plant and female papaya plant, and they are also having separate flower. Male flowers are very different from the female flower, and female flowers will lead to formation of papaya fruit. That's why papaya fruit is growing in female plant. A better diagrammatic representation of flowers of papaya plant, male flower is having unique morphological structure with reference to the female flower of papaya plant. Oh, the sixth point, why I've written it as fifth point. The sixth outbreeding device is self-incompatibility. Self-incompatibility is a genetic mechanism which usually prevent self-pollination to occur, and it helps in cross-pollination, and thereby there is pollen germination of external pollens. Say in case of this ovary having genetic genotype of S1 and S2, there is no fertilization of S1 and S2 pollen grains. There is no pollen germination of S1 and S2. Instead of that, there is germination of external pollen grains having different genotype. So there is pollen germination of S3 and S4 type of pollen grains as the genetic makeup or genotype of the pistil is S1 and S2. So a pistil having S1 and S2 genotype will not allow the S1 and S2 type of pollen grains to germinate. Instead of that, it favors germination of pollen grains having different genotype, different gene. So S3 and S4 will be germinating and growing pollen tubes inside the style of a pistil having S1 and S2. And there is prevention of germination of pollen grains having S1 and S2 genotype or gene in case of a pistil having S1 and S2 genotype. So these are six. This number should be 6. I'll change it later. So the seventh outbreeding device that we are going to discuss is Polenprepotency. Prepotency is an ability of external pollen grains to germinate on a stigma of pistil. Usually a stigma of pistil doesn't allow the self-pollens to germinate. Say in case of this pistil which is having S1 and S2 type of genotype will not be allowing a self-pollen having S1 and S2 genotype to germinate and grow pollen tube, but it do allow external pollen grains to germinate faster compared to the self-pollen. So this is usually called as Polenprepotency, hence doesn't allow self-pollination to occur. Instead, it is favoring cross-pollination. S3 type of pollen grain is usually an external pollen grain which was cross-pollinated due to pollinators. So these are the different outbreeding devices which we had discussed. The number should be 7, right? So first outbreeding device that we had discussed is dichogamy, that is maturation of pollen and ovary at different times. And second, heterostyly, difference in the length of style of the pistil. And third one, dicliny, which is occurrence of male flower and female flower, or growing of male flower and female flower separately on plant. And then the fourth one is dioecy, presence of male plant and female plant having their own pistillate and staminate flower. And fifth one is self-incompatibility, which is not allowing self-pollen to germinate and allowing foreign or external pollen grains to germinate. And the last one that we had discussed is Polenprepotency, and we haven't included here with the Herkogamy, right? Herkogamy include heterostyly. So now let's discuss about artificial hybridization of plant. What is artificial? Artificial here refers to involvement of human manipulation. Human is trying to manipulate plant, and hybridization is a process of formation of hybrid plant from two parents, superior parent. So artificial hybridization is breeding between genetically dissimilar plants, which usually produce a superior hybrid having superior genetic combination. So the hybrid which is produced by hybridization of plant usually have superior genetic combination, which in a way is superior plant. It can be artificial hybridization can be done between two different variety, that is called as intervarietal hybridization. Interspecific hybridization is done between two different species, and intergeneric hybridization is carried out between two different genera of plant. To make it more clear, I have a few card animations here. So suppose if we have a superior pea plant having high protein quality or high protein content, if we cross or interbreed or hybridize it with another plant which is resistant, disease resistant to pathogen, then there is production of hybrid plant which is having high protein quality. Along with that, the plant is more resistant to external pathogens. So in a way, there is production of hybrid plant which is having ability to survive more, and which usually provide our seeds having high protein quality. So this in this way, by selecting two superior parent plant, there is production of hybrid plant which is having superior genetic combination and is having better quality and quantity production of food, seed, is what is artificial hybridization of plant is. So the main objective here you can draw out is mainly for improving the crop variety. If we are able to produce different crops, then there is we are indirectly securing our food. So for example, we have different rice, different types of rice: basmati with sweet, good smell, and local. We usually have rice having high starch and rice having high protein content. So all these diverse different rice usually secure food for future. Along with that, we are indirectly creating or producing rice seeds which are more having more ability to survive, which are more viable. So the main objective of artificial hybridization is to improve crop variety. Now these techniques usually make sure that desired pollen grains are being transferred, and protection of stigma from contamination by unwanted pollen grains. So these 2 points are usually kept by the scientists or the farmers or the gardeners who are practicing artificial cross-pollination. So the main procedure or protocol which is involved in artificial hybridization of plants are emasculation and bagging technique. These 2 techniques are important in artificial hybridization, though there are many steps involved in artificial hybridization, regarding which we'll be studying in another chapter in say strategies for enhancement of food is a chapter under which we'll be discussing in detail about artificial hybridization. Over here we'll discuss about emasculation and bagging technique. So in case of bisexual flower, the artificial hybridization that we usually practice is emasculation. So the main technique is emasculation. E refers to removal. Masculation here refers to anther, the male part of the plant. Mainly the anther which produces pollen grain. So removal of anther. So anther is removed when the flower is at bud stage. So when the flower is at bud stage, by human disturbance, or in a way scientists will be manipulating the flower by removal of immature anthers. Before the maturation of anthers are removed with the help of forceps. After removing anther, there is no possibility of self-pollination occurring, so that we can dust out desired pollen grain onto the stigma and we can get the desired plant, right? So the second technique that we had to discuss is bagging. Bagging, the literal meaning itself says that covering up. So to this flower we usually cover a suitable bag, which is usually made up of butter paper. So this this technique of covering up of the emasculated flower is mainly to protect the stigma from undergoing pollination from unwanted pollen grains. Now this protected flower will be later dusted with the desired pollen grains having superior quality with superior genetic combination, and after dusting it, then again it is covered with butter paper, and which in a way allows cross-pollination, which in a way allow the desired pollen grains to germinate and grow pollen tube, which later leads to formation of seeds after fertilization. And in this way we are able to produce seeds, and seeds on germination we are producing hybrid plant with superior quality. So this is in this context I've written what I had explained. So as the stigma of the bag of flower attains receptivity, then desired pollen grains which are collected from anther of desired main plant are dusted on stigma, and then flowers are rebagged as we have seen in animation, and finally fruits are allowed to develop. Inside fruit will be having seeds. So seeds on germination we are able to reproduce the hybrid plant. So this is how artificial hybridization is done in case of bisexual flower. Now in case of unisexual flower, flowers are only the female flowers are only having stigma and there is no presence of anther. And do you think emasculation is required? There is no need of emasculation. Anthers need not be removed, as the flowers are only having the pistil with stigma. So what it need to be done is the female flowers should be protected from pollen germination, unwanted pollen germination. So for that we use butter paper for bagging, and once once the flower attains maturity and after collecting the desired pollen grain, then pollen grains are dusted. So once the stigma becomes receptive, then we are dusting pollen grains, and then we are rebagging it. And after rebagging it, there is formation of seeds, and seeds are formed inside fruit, and seeds on germination we are able to reproduce another unisexual plant, right? So this is how unisexual flowers are artificially hybridized. In case of unisexual flower, there is no need of emasculation, as the flowers are only having the female reproductive part, that is pistil. So with this we have come to the end of this video. You need to answer these questions. The first question is, what are outbreeding devices? Flowering plants opt for outbreeding devices though it is uneconomical. You have to give reason for this. In the third question, you have to discuss at least 5 outbreeding devices with an examples each. And fourth, you have to explain artificial hybridization in bisexual plant. Fifth, how artificial hybridization in bisexual flowers is different from artificial hybridization in unisexual flowers. So you have to draw out the differences between artificial hybridization in bisexual flower and in unisexual flower. And in the sixth question, you have to mention the objectives of artificial hybridization. In the seventh, you have to describe emasculation with reference to artificial hybridization. With this, we'll stop this video, and I'll see you in another video. Till then, take care of yourself and keep smiling. Bye.